Hi everyone, my name is Ashby and I'm a senior at Pomona College, and I will be presenting the research I conducted this summer at Memorial Sloan Kettering Cancer Center in conjunction with Dr. Ronglai Shen and Dr. Saptarshi Chakraborty. I want to begin by focusing your attention on this word cloud on the right. This word cloud was generated from the Cancer Genome Atlas webpage. A key insight you can glean from this graphic is that green words outnumber the large multicolored words, indicating that text data is dominated by rare words. This is also true in the language in which cancer operates, the language of somatic variant mutations. So finding ways to harness the vast array of rare mutation for important clinical tasks is an important statistical and biomedical endeavor. The specific clinical task that we're trying to address is diagnosis of cancers of unknown primary site. 3 to 5% of cancer diagnoses globally are of unknown tissue of origin, and identifying a cancer's primary site is a critical step in informing specific, effective, and minimally toxic treatments. Since mutation data is dominated by rare mutations, if you sequence a new tumor sample, you're bound to encounter some mutations you've never seen before. Let's say you're trying to decide whether this tumor, with a previously unseen mutation shown in the red bold X at a particular locus along gene 1, let's say you're trying to decide if this tumor originates in tissue A or tissue B. If you look at the mutations in gene 1, cataloged in previous tumor sequences under tissue A and B, you notice that gene 1 has a greater diversity of mutations in tissue type B. So encountering a new mutation in gene 1 is more probable within the context of tissue B. Thus, it is more likely that our new tumor originated from tissue B. We can rigorously generate probabilities of encountering previously unseen mutations in major human cancer genes across different tissue contexts using a Bayesian nonparametric method called good Turing frequency estimation. When we apply this method to 7,000 tumor samples in the Cancer Genome Atlas, we see remarkable variation in these unseen variant probabilities, as shown here in this bubble plot. On the x-axis are 32 different primary sites, and on the y-axis is the gene label for the gene VHL, and the bubbles are sized based on the probability of encountering a previously unseen mutation in a future sequenced tumor of these particular primary sites. So this heterogeneity in these unseen variant probabilities can be leveraged for tasks in primary site classification. However, this method was only applied to major cancer genes, around 300 out of the 20,000 total genes in the human genome, due to mutation data sparseness. We asked the question of whether we can extend this analysis to a larger fraction of the cancer exome by considering groups of genes. We propose a thoughtful way of grouping genes using biological features known to influence somatic mutation rate, and we term these features metafeatures. These are the metafeatures that we looked at. We looked at a gene's average expression across 91 cell lines, each gene's replication time, each gene's non-coding mutation rate, each gene's GC content, and each gene's presence in an open or closed chromosomal compartment. And so the intuition of our approach is we're zooming out on the genomic landscape and considering groups of genes built to resolve mutation data sparseness while still retaining the tissue-specific signals encoded by the rare variant probabilities. We built these gene groups using a k-means approach with different random initializations of our cluster centers. The height of the cloud of pink points corresponds to the tissue specificities of our unseen variant probabilities generated from 10,000 metafeature-based clusters. We compared the tissue-specific signals of our clustering approach to the signals of null or random groupings, and the quantiles of those tissue specificities are shown in the multicolored lines. The slight boost in the pink cloud indicates the metafeatures carry some signal, although not enough to exceed the null distribution produced by random groupings. So in summary, the punchline of this work is we've developed a framework to resolve mutation data sparseness and yield tissue-specific signals by grouping genes using metafeatures. The most immediate next step is that our metafeatures are likely too crude to significantly improve upon a random grid search approach, and we need to sophisticate our metafeature space to include more tissue-specific metafeatures. And down the line, we hope to identify highly tissue-specific gene groups of high diagnostic value and use them to build a classifier of cancer primary site. Thank you for listening. If you have any questions, please ask them below in the chat box. Thank you.